Unit 3 Cell Structure and Functions. Chapter 8 The Unit of Life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvements leading to the electron microscope revealed all the structural detail of the cell. Cell theory. In 1838, Matthias Caldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plants. At about the same time, Theodor Swan, 1839, a British geologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Swan proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. Caldon and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, omnicellula is cellula. He modified the hypothesis of Caldon and Swan to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and product of cells. All cells arises from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel and a human cheek, cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack membrane-bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cells. Various chemical reactions occur in it to keep the cells in the living state. Beside the nucleus, the eukaryotic cell have outer membrane-bound distinct structure called organelles like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on rough air. Animal cells contain another non-membrane-bound organelles called centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human RBC are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like polygonal-like, columnar, cuboidal, thread-like or even irregular. The shape of the cell may vary within the functions they perform. Prokaryotic cell. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cell. They may vary greatly in shape and size. The four basic shapes of the bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and xylem, spiral-shaped. The organization of the prokaryotic cell is fundamentally similar even though prokaryotes exhibit a wide variety of shape and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filled in the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome, circular DNA, many bacteria have small circular DNA outside the genomic DNA. The smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of enclosures. A specialized differentiated form of cell membrane called mesosome is the characteristics of prokaryotes. They are essentially unfoldings of cell membrane. Cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layer structure that is the outermost glycocalyx followed by the cell wall and the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in cell envelope and the manner in which they respond to the staining procedure developed by Gram, where those that take up the Gram stain are called Gram-positive and others that do not are called Gram-negative bacteria. Glycocalyx differ in composition thickness among different bacteria. It could be a loose sheet known as slime layer in some, while in others it may be thick and tough known as capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside the world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contains pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and the arrangement of flagella. Bacterial flagellum is composed of three parts: filaments, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili, fimbriae are also surface structure of the bacteria but do not play a role in motility. The pili are elongated tubular structure made up of special protein. The fimbriae are small, bristle-like fibers sprouting out of the cell. In some bacteria, they are also known to help attach the bacteria to the rock stream and also to the host tissues. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA into protein. Inclusion bodies. Rhizome materials in prokaryotic cells are sorted in the cytoplasm in the form of inclusion bodies. These are not bounded by any membrane system and rise free in the cytoplasm. Example, phosphate granules, cyanophysin granules, and glycogen granules. Gas vacuole are found in blue, green, and purple and green photosynthetic bacteria. For second part के लिए आप नेक्स्ट वीडियो में जाके देखिए. थैंक यू सो मच.